गुड इवनिंग बच्चों, वापस तो हम अब सेकंड पार्ट की बात करने वाले हैं. वी आर गोइंग टू टॉक अबाउट द सेकंड पार्ट ऑफ माइक्रोब्स इन ह्यूमन वेलफेयर, दैट इज योर use of microbes in industry. Means use of microbes at industrial level. Now we are going to see. Okay, so in the previous lecture we studied about that different use of microbes in household. So first we have to revise that thing. Okay, so in that first part we are discussed about your 5 to 6 microbes. The first one is your Saccharomyces cerevisiae, which deals with toddy and your bread. Okay, second one is Penicillium roqueforti, which deals with Roquefort cheese. Okay, third one is your Lactobacillus acidophilus, which deals with your idli dosa and curd, and Penicillium camberti with your camembert seeds, and your Papillobacterium shermani with your Swiss cheese, and that Roquefort cheese with your Roquefort, that Penicillium roqueforti with Roquefort cheese. Okay, so these all are different microbes which we studied previously in the chapter related to that household. Okay, now this second one is related with the organisms which is used in industries. On industrial level now we are going to talk. Okay, so in industries microbes are used to synthesize a large number of products which are valuable to human. Okay, in industry we are use microbes in very large level. Okay, now we are not talking about for a single family. Now we are talking about whole population. Okay, so in this part we are talking about the use of microbes on very large scale, and it is for human welfare. Okay, so let's get started. So the first thing which we are going to study in this chapter, in this part, is that fermenters. Okay, for the production of products at industrial scale there is requirement of fermenters. Okay, when you are going for a race, you need a car. Okay, when you are going to play cricket, you need bat and ball. Okay, simultaneously when you are doing this use of microbes in very large level, then you require fermenters. Okay, fermenters is that thing in which you will increase the quantity of microbes. You have a small amount, you add that thing in fermenter, it will increase the quantity. Okay, just like khulja sim. Okay, in which you add a magic and the thing is enlarges. Doraemon la big torchlight, just like this. Okay, so for the production of products at industrial scale there is requirement of fermenters. In fermenter large number of microbes grows. Many fermenter combines to form fermentation plants. Okay, when you have many fermenters, 100 to 200 fermenters simultaneously is working in a factory, in an industry, then that is called fermentation plants. Okay, these are the fermenter, and this is the whole fermenter plant where different types of fermenter working together and doing and producing that material you want on industrial level. Okay, so first microbes are in this production are various industrial products like beverages, pharmaceuticals and chemicals. Okay, in this industrial level we produce beverages. Okay, we produce medicines, antibiotics by the help of this method, and chemicals. Acids, enzymes are produced by the help of microbes in large level. That is the use of microbes in industrial level. Okay, so microbes, especially yeast, have been used for the production of beverages like beer, wine, brandy, rum and whiskey. That thing is called brewer's yeast. Again, species is same, Saccharomyces cerevisiae, and now its name become brewer's yeast. Okay, so the yeast used in this process is Saccharomyces cerevisiae. It is also known as bread making. It is commonly called brewer yeast. Okay, so this is your yeast, and we produce these things from this. Rum, whiskey, beer, wine, brandy, all these are produced in the distilleries, in the fermenters by the help of Saccharomyces cerevisiae, that is your yeast. Okay, so the raw material is fermented with the yeast break down the raw material and produce the specific product. Okay, this is the fermenter. You put raw material inside the fermenter and you put yeast inside that. Yeast halfly digests that, okay, inside that anaerobic condition and produce what? Ethanol. Okay, so fermented beverages, in the process glycolysis, glucose is the raw material and converted into pyruvate to release ATP. Okay, that is a normal thing that you studied in respiration in plants. Okay, first step of alcoholic fermentation, pyruvic acid is decarboxylated. It loses the molecules of carbon dioxide. Means from your glucose you are removing CO2. Okay, as a result, acetaldehyde is formed, which is a 2 carbon molecule. When you remove that thing from your pyruvic acid, when you remove the carbon dioxide, pyruvic acid, it is 3 carbon compound, and you removing what? CO2. CO2 is 1 carbon compound. Okay, now you remove 1 carbon from that 3 carbon compound, then it is become a 2 carbon molecule, which is called acetaldehyde. Now NADH formed during glycolysis gives its electrons and hydrogen to acetaldehyde, and this regenerates NADH. Okay, and the end of ethanol and CO2 are the product. That is the common reaction. Okay, here you can see pyruvate, a 3 carbon compound. By the help of enzyme pyruvate decarboxylase, you will convert this thing into acetaldehyde, and the enzyme and the process is called decarboxylation, means removal of CO2 from a compound. That process is called decarboxylation. Okay, so pyruvate is converting your acetaldehyde, C3, a 3 carbon compound, is converting into 2 carbon compound acetaldehyde. Now this acetaldehyde will convert into ethanol by the help of enzyme alcohol dehydrogenase. Okay, alcohol dehydrogenase is used to convert your acetyl CoA, that acetaldehyde into ethanol by the help of enzyme alcohol dehydrogenase, and you get the end product, a 2 carbon compound ethanol. Okay, so it is the mechanism, it is the basic idea how your ethanol is produced. Okay, so now from where we are, the raw material. What is the raw material of ethanol? So for beer it is wheat. Okay, for wine it is grapes. Sugarcane molasses for rum. Okay, barley for whiskey, and potato peels is your vodka. Okay, again I am saying beer is produced by wheat molasses, then grapes is used to produce wine, then sugarcane is used to produce rum, while your barley is used to produce whiskey. Okay, and potato is used to produce vodka. So these all are different raw material used to produce different alcoholic beverages. Okay, so the production of alcohol take place by fermentation and distillation. Okay, so first we do fermentation. Okay, after fermentation, to increase the concentration of alcohol inside the medium, we do distillation, and after that we drink that. Okay, now on the basis of that, we have two types of drinks: distillated drinks and non-distillated drinks. Non-distillated drinks are those drinks which are not distilled. Okay, see. So distillation is your rum, whiskey, vodka, okay, champagne, okay. Non-distill is your wine and beer. Okay, so distilled drinks, distilled alcoholic beverages is your rum, whiskey, vodka, champagne, while your non-distilled thing is wine and beer. Okay, now we will see what is this distillation. Okay, so distillation is the separation and removal of alcohol from other components from alcoholic beverages. Okay, you are removing that separation and removal of alcohol from other components from alcoholic beverages, means that material that is present inside the drink, you have to remove alcohol from that thing. That process is called distillation. Removal of alcohol from other things from that alcoholic beverage is called distillation. Okay, in distillation the alcohol is first evaporated and then allowed to cool. Okay, first you make it evaporate. After evaporate, it will get precipitate, and then it can condense, and you will store that thing. That thing which is coming out in the form of water vapor and condensing and accumulating in other container, that is your alcohol, and that is your distilled form of alcohol. Okay, since alcohol has a lower boiling point, it boils first and turns into the vapor, and these vapors are then condensed in a separated vessel. Okay, first you evaporate that thing. Okay, as you know that alcohol has very low boiling point. Okay, so it first evaporate, it get first boil, and then it will condense, and you will accumulate that thing in other container. Okay, that's how you do distillation. Okay, this result in separation of alcohol from the component, the raw material which is present, which you do not want, is being thrown out, and the thing which you want, that is alcohol, is being stored in different bottles in different containers. Okay, that is the process of distillation. So depending on type of raw material used for fermentation, there are type of processing with or without distillation. Different types of alcoholic drinks are there. Wine and beer are produced without distillation. Whiskey, brandy, rum are produced by distillation. Again, I am saying beer and wine is produced without distillation. Whiskey, rum, vodka is produced by the process of distillation. Okay, so this is your distilled drinks, brandy, rum, whiskey, and this is non-distilled, beer and wine. Okay, vodka is also a distilled drink. Okay, so but at last, alcohol is not good for health. Okay, drinking alcohol is hazardous to your health, hazardous to your liver. Okay, so what are the negative points of alcohol? Okay, aggressive behavior. Okay, drop in academic performance, lack of interest in personal hygiene, stealing to be able to buy these are very costly things. Okay, so some people start doing stealing things, robbing the houses. Okay, so these all are the negative points of drinking alcohol. And on body, it will do what? Increase urine output, diuresis. Okay, it will affect your aldosterone, ADH hormones inside the body, and due to which over urination take place, that is your diuresis, and it will cause dehydration inside your body. Okay, second, liver disorder like cirrhosis of liver, your liver become fatty liver. Okay, because at a limit is there that liver can detoxify things, but if get it over, then it will convert into cirrhosis. Your liver get cirrhosed, means your damage of liver get started. Okay, so these all are the bad thing of alcohol on the body. Effects on brain. Okay, your neural system get affected mostly, and on your brain it will affect your cerebellum. You cannot move properly, your voluntary movements get affected. Okay, so these all are the negative effects of alcohol. So say no to drugs, say no to alcohol. Okay, so next use of microbes on industrial level is antibiotics. Okay, so chemical substances which are produced by some microbes and can kill or inhibit the growth of other disease causing microbes is called antibiotic. Okay, now in biology every word sees its meaning. Okay, so antibiotic, we break this word. Anti means against, biotic means living. A thing which grow against the living thing, a thing which avoids the grow of living thing is called antibiotic. Okay, so anti against biotic life, a thing which is against the life is called antibiotic. Okay, so anti is a Greek word that means against, and bio means life. Together they means against the life in concern to disease causing organisms. Okay, and this against the life is for disease causing pathogen, not for normal life. Okay, it is because it is towards the pathogens which causes disease in the organism. Okay, so whereas with reference to human being they are pro life and not against. Okay, for us, for human content, they are not against the life, they are pro for life, they are life giver, they are avoiding the disease inside the human body. Okay, so this is your Alexander Fleming. Okay, the person behind the discovery of antibiotics, the person who first discovered the antibiotic. Okay, so antibiotics are the medication that can kill bacteria. Antibiotics were discovered by Alexander Fleming. Okay, and he discovered what? Penicillin. Listen one thing, penicillin is antibiotic and penicillium is the organism. Okay, Okay. So Alexander Fleming was working on Staphylococcus bacteria in his lab. He observed a mold growing in one of the unwashed culture plate where Staphylococcus could not grow. Okay, this discovery of penicillin is accidental. It is not a real discovery. Okay, वो जबरदस्ती करी हुई डिस्कवरी नहीं है, वो इंटेंशनली करी हुई डिस्कवरी नहीं है. It is an accidental discovery. Okay, when this guy Alexander Fleming working in his lab on a bacterium staphylococci, okay, he find that in a unwashed petri dish some colonies are growing and it's secreting out some chemicals that inhibiting the growth of staphylococci bacteria. And after the studies or microscopic studies on that, he find that it is a fungus from your Ascomycetes class, that is your penicillium. Penicillium notatum is the fungi which is responsible for the production of antibiotic penicillin, which is killing this bacteria inside that petri dish, which is accidentally discovered by your Alexander Fleming. Okay, so he observed that mold growing in one of the unwashed culture plate around which staphylococci could not grow. He found out that it was due to the chemical produced by the mold. Okay, he named it penicillin after the mold, Penicillium notatum. The species which is responsible for the production of penicillin is Penicillium notatum. Okay. So antibiotics, Ernest Chain. Now after that, two more scientists come. That is your Ernest Chain and Howard Florey. Okay, they found full potential of penicillin as effective antibiotics, and this antibiotics was extensively used to treat American soldier wounded in World War II. Okay, these two are the Ernest Boris Chain and Sir Howard Walter Florey. Okay, these two guys advance, modify the power of penicillin, and it is used during the World War II by American soldiers. Okay, so these three guys, Fleming, Chain, Florey, were awarded Nobel Prize in 1944 or 1945 for the discovery of antibiotics. Okay, Sir Alexander Fleming, Ernest Boris Chain, and Sir Howard Walter Florey get award Nobel Prize in 1945 on the discovery of penicillin. Okay, this guy discovered the penicillin, and these two guys have modified and initiate the more powerful powerness of your penicillin. Okay. Next, antibiotics. After penicillin, other antibiotics were also purified from microbes, just like your Cephalosporium acromonium. Okay, it's technically cephalosporin. Okay, next, Bacillus subtilis, bacterium. Okay, then Streptomyces garius, streptomycin. Okay, Micromonospora purpurus, that is gentamicins. All these all are bacteria, and this one is your that cephalosporin is also produced by microbes. Okay, so this Cephalosporium archaeomonium, a bacterium which produces cephalosporin. Okay, then Bacillus subtilis produces bactericin. Okay, Streptomycin gralius, streptomycin, and Micromonospora, that purpura, that produce gentamicin. Okay, so these all are the more antibiotics discovered by us after the discovery of penicillin. Okay. Next, antibiotics have greater improved our capacity to treat deadly diseases like plague, diphtheria, whooping cough, kali khasi, leprosy, kushth rog, diphtheria, galghotu. All these are very dangerous bacterial diseases which can be easily treated by these antibiotics. Okay. Next, largest antibiotic producing genus is your Streptomyces. Okay, did you know largest antibiotic producing genus is your Streptomyces? Okay, Streptomyces is the largest antibiotic producing genus in the microbial world. Streptomyces is a genus of fungus. The species are responsible for the product producing maximum number of antibiotics. The synthesis, the antibiotics which have a wide range of activity like being antibacterial, antifungal, and antiparasitic in nature. They also know that produce certain compound which can act as an immunosuppressive agents. Okay, so these all are your Streptomyces and fungus which is responsible for production of many different antibiotics and largely used to produce antibiotics. Okay, then chemicals, enzymes, and bioactive molecule which is produced with the help of microbes. Okay, so first we have chemicals, any material which is definite chemical composition that we call chemicals. Enzyme, substance which is a biological catalyst is an enzyme, and bioactive molecule which are produced as a result of microbial activity. So these all are chemicals, enzymes, bioactive molecules are also produced by your microbes. Now we are going to see one by one different chemicals, enzymes and bioactive molecules. Okay, so first we have organic acids, that chemicals. Okay, so first we have citric acid produced by Aspergillus niger, a fungus. Okay, and it is used to produce detergent, preservatives, flavoring agent, leather tanning. Okay, so these all are the functions, that are all the uses of your citric acid, that is detergent manufacturing. Okay, preservative, flavoring agent and leather tanning. Okay, so these all are the uses of citric acid, and it is produced by your Aspergillus niger, which is a fungus. Okay, second organic acid, second chemical which is produced by the help of microbes is your acetic acid. Okay, and it is used as industrial solvent. Okay, it is used as a solvent. Okay, cancer treatment and food. Okay, acetic acid produces by a bacterium, that is your Acetobacter aceti. You know vinegar, in vinegar you will find Acetobacter aceti. Okay, next one is your organic acid is butyric acid. Okay, used as a food and flavoring, that flavoring agent. Okay, pharmaceutical and perfumes. Okay, this butyric acid is used in food and flavoring, and your use in your use in producing in the drugs. Okay, pharmaceuticals. Okay, and perfume. Okay, so the organism which is used to produce this organic acid, butyric acid, is Clostridium botulicum. Okay, it is again a bacterium. Next organic acid is your lactic acid. Okay, cosmetic industry it is used, pharmaceuticals it is used, food industry it is used. Okay, so it is produced by a bacterium, that is your lactobacillus. Okay, the lactobacillus strain, like the lactobacillus genus, is used to produce this lactic acid as an organic acid, as a chemical. Okay, next alcohol. Okay, you know alcohol. Okay, alcohol, it is also produced by your Saccharomyces cerevisiae yeast. Ethanol, it is used in food industry, pharmaceuticals to produce sanitizers. Okay, cosmetic industries to use as a base in cosmetic industry. Okay, next one is your enzymes. Okay, now enzymes are also present, which is produced by the help of microbial activity, and it is also used in daily life. Okay, so first we have lipase. Okay, so lipase are used as a detergent formulation and helpful in removing oil stains from the laundry. Okay, when your clothes get dirty, you will wash them with the help of detergent, and you know that detergent contain lipase enzyme, and it is also extracted out from microbes. Okay, next one is your pectinase and protease. Okay, you know you have two types of drinks in the market. One is the pulpy one, and another one is clear drinks. Okay, that pulpy drinks does not contain these pectinase and protease, but the clear drinks, just like fruity, maza, they contain pectinase and protease. That's why the drink is clear, it does not contain any pulp. Okay, next one is your streptokinase, very important enzyme from the point of meat also and from your boards also. Okay, so it is produced by bacterium streptococcus and modified by genetic engineering to be used as a clot buster in patients who have undergone myocardial infarction. See what this thing do. Streptokinase is an enzyme which is produced by organism Streptococcus bacterium. This thing that blocks the thing which is blocking the clot, which is blocking the arteries of our heart, has been blasted down by the help of this enzyme streptokinase. It will bind with the clot and destroy the clot, that blood can easily flow down inside the artery. That's how it's work. Okay. Next, the enzyme pectinase and protease are used to clarify commercial fruit juice by cleaving pectin and the peptide bonds respectively. Okay, this is your homemade juice. You add pectinase and protease, it will become clarified bottle juice. It look like your clarified bottle juice. Okay, so enzyme, that is your streptokinase. Okay, so chemical, enzyme and biochemical activity. Enzyme, myocardial infarction, plaque rupture and causes blockage, damage and death of cardiac muscle can happen. Okay, so now the function, the patient who having these conditions, okay, in which plaque rupture and causes blockage, damage and death of cardiac muscle, in that case the injection of streptokinase has been injected near the chest region. It directly go inside the circular, that's a systemic circulation, okay, and blast down the clot. Okay, the clot, the blockage which is taking place inside the main arteries, okay, it been blocked down, it been blast down with the help of this enzyme streptokinase. Okay, remove blood clots from the patient who have undergoes myocardial infection like this. Okay, this is a block caused by, now this blood cannot move ahead. Okay, you can see here, this is the blockage produced inside the that artery or capillary. Now this blood cannot move here, so now the tissue from here start dying because of hypoxia. Okay, now what will we do? We will inject the injection of streptokinase. Now what will happen? Your streptokinase will go and open this clot. Okay, blast down this clot. Now your blood can move ahead, and the area where the oxygen is not reaching, it get oxygenated, you survive. Okay, next bioactive molecule, okay, that is your produced by Trichoderma polyspermum, is a fungus which produce a bioactive molecule which is your cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients. Okay, so what is immunosuppressive agent? Okay, these agents are injected inside your body to depress down your immunity, to make your immunity go down, okay, during the time of that organ transplant. Because in organ transplant what happened, you will transplanted organs from some other donor. Okay, so it can be happen that your body cannot take this organ, okay, does not recognize that organ. Okay, so what will your body do? It will send its antibody to kill, to destroy that organ. So at that time we'll low down that immunity of our body by injecting this cyclosporin A to low down your immunity, and that your body can easily mix with that organ. Okay, this thing also used in the conditions like your autoimmune disorder, in which your own immunity start to killing your body cells. Okay, step that cyclosporin A is also used in the conditions like your autoimmune disorder, where immunity is stand against your own body. Okay, so this is your Trichoderma polyspermum, a fungus which is producing cyclosporin A, and it is used as a immunosuppressive agent which decrease your immune power. Okay, immunosuppressant allow acceptance of donor organ. See, this is the living donor and this is the recipient. Okay, immunity goes down during the installation of this immunosuppressing cyclosporin A, and you can easily donate the organ inside the recipient body. Okay, next bioactive molecule is statin, produced by yeast Monoscus purpurus. हर yeast का scientific name Saccharomyces cerevisiae नहीं होता. ओके, यहाँ पे तुम्हारे पास एग्जांपल है. तो स्टेटिन ये बनाता है एक ईस्ट जिसका नाम है मोनस्कस परप्यूरस. ओके, दे आर यूज्ड एज अ ब्लड cholesterol lowering agent. Okay, you know today food style. Okay, you are eating that fast food which is full of junk. Okay, it will accumulating inside your arteries. Okay, so cholesterol level is increasing inside the body. So doctors give medicines which break down, which destroy this cholesterol. Okay, so this is a class of drugs which inhibits to accumulate cholesterol inside the body, which destroy the cholesterol which is installed inside your body. That category of drugs are called statin, and it is produced by a yeast species which is called Monoscus purpurus. Okay, they act by competitively inhibiting the enzyme responsible for the synthesis of cholesterol. Okay, it will do what? It will go and bind to the place where the cholesterol formation take place. So the enzyme which is responsible for the production of cholesterol inside the body does not get that enzyme, and on the place of that enzyme, that statin will bind, and the cholesterol formation does not take place. I have a medicine for that, just like this. This is your ateroliptan. Okay, it is also an statin drug which contains this thing. Okay, you can see here. Okay, this is your ateroliptan. 10 mg of statin this drug contains. Okay, So I have a question. A patient brought to a hospital with myocardial infarction normally immediately give what drug? You will give cyclosporin A, streptokinase, penicillin, statin. What will you do? What will you give? Okay, let's discuss this question. Okay, cyclosporin A. This is your immunosuppressant. So you are not going to give this. Okay, then penicillin, antibiotic. Again, this thing you will not give to that patient who having myocardial infarction. Okay, statin, that is your cholesterol lowering agent. Again, you will not give this. Okay, because it is a cholesterol lowering agent, does not affect that blockage of heart, myocardial infarction. Then what will you give? You will give streptokinase. Okay, so correct answer is streptokinase. Okay, streptokinase is given to a patient who is brought to the hospital with myocardial infarction. Okay, you will inject down streptokinase injection in the upper chest region, this rear, okay, near to the heart. It will directly hit your heart and then it will absorb inside the body and destroy the clot which is happened, which is produced inside the heart, inside your body. Okay, so now what we studied till, okay, summary time. Okay, so we have studied about fermented beverages like beer, wine, brandy, rum, whiskey. Out of that, beer and wine are non-distilled and your brandy, rum, whiskey is distilled, and this is produced by Saccharomyces cerevisiae by the process of alcoholic fermentation on raw material. Also we studied that raw material, wheat is used to produce beer, sugarcane molasses is used to produce your rum, okay, while your that potato is used to produce vodka, while your grapes is used to produce wine, okay, and other substances, other rye, molasses is used to produce your whiskey. Okay, second, antibiotic we studied. Okay, so these all after penicillin, other antibiotic were also purified and developed from other microbes. So we studied that cephalosporin, acromauronin, this produced cephalosporin. Okay, then Bacillus subtilis is produced bacteriostatin. Then Streptomyces graecis it produced streptomycin, and Micromonspora purpura produces gentamicin. Okay. So organic acid we studied in this part. Okay, lactic acid produced by lactobacillus. Okay, then your butyric acid produced by Clostridium butyricum. Then acetic acid produced by Acetobacter aceti, a bacterium, and then citric acid produced by Aspergillus niger, a fungus. Okay, so these three are bacteria: lactic acid by bacteria, butyric acid by bacteria, acetic acid by bacteria, citric acid by fungus. Okay, and this is the whole that summary of the chapter that we studied till. Okay, lipase, okay, use is for detergent, pectinase, protease, used to clarify bottle juices, streptokinase, an enzyme produced by streptococcus, used as a clot buster for myocardial infarction. Then we have two bioactive molecules, cyclosporin A and statin. Cyclosporin A is produced by a fungus species, Trichoderma polyspermum, that is an immunosuppressive drug use of that. Statin produced by a species of yeast, Monoscus purpurus, work is blood cholesterol lowering agent. Then we have alcohol, ethanol produced by Saccharomyces cerevisiae, that is your brewers yeast, production of alcohol drinks and disinfectant. Okay, now in next lecture we are going to study about microbes in human welfare, in which we are going to discuss biogas production and biocontrol. Okay, thank you for now. That is for today. Okay, after tomorrow or in the evening class we will discuss about that biogas production and biocontrol agent. Okay, endigu then digu nama tapasya and kabhi jhuknega nahi. Okay, thank you.